Hello, everyone, and thank you all for signing up and attending our second IYCN Science Café. This Science Café will be with the title Chemical Synthesis of RNA and DNA. We would like to thank Professor Ely for accepting our invitation to give this talk. I will be with you. My name is Witwatersrand, and I am IYCN's Conference President's Committee Co-Chair, together with my colleague Tracy Lowe. Tracy, introduce yourself.
Thank you, Walid. My name is Tracy Lau. I am the IYCN Conference President's Committee Co-Chair as well. Thanks, Walid.
Okay. We will begin with a brief introduction about the IYCN, and then followed by it will be a 30-minute talk with, by Professor Abdullah, and then we will have 20 minutes Q&A session and 5 minutes for a closing remark. First, I would like to tell you more about our organization. We are the International Younger Chemists Network. We are an associated organization of the IUPAC since 2017. Our vision is to connect and empower younger chemists globally, and our mission is to support and advocate younger scientists across the chemical sciences toward a globally sustainable future. For those who want to join the IYCN committee, you can sign up through this link, iycnglobal.com/membership. The membership is for free. You just need to be pursuing a degree in chemistry related degree, and you need to be under 35 years. This is a photo of our delegates in the first IYCN Global Assembly in Paris 2019. For those who want to connect with us more, you can follow us on multiple social media platforms like LinkedIn, Facebook, Twitter, and Instagram. Now, for a brief introduction about our speaker, Professor Abdullah Ely is an associate professor within the Antiviral Gene Therapy Research Unit at the Faculty of Health Sciences at the University of Witwatersrand, South Africa. He completed his PhD in 2009, which focused on using the RNA interference pathway for targeted silencing of hepatitis B virus, HBV. Since then, he focused on the development of novel gene therapy technologies against HBV. More recently, his research has explored the use of in vitro transcribed mRNA for the development of antiviral therapeutics as well as vaccines. Without further ado, Professor Ely, you can share your screen. Thanks.
Thank you, Walid, and thank you for this invitation. It's a pleasure to be here, and I hope you will enjoy the presentation that I've put together. Oops, let me just quickly put off my webcam.
Okay.
Okay, so yeah, again, thank you for the introduction and thank you for the invitation to present our work. As Walid has said, I am Abdullah Ely, and I am from the Antiviral Gene Therapy Research Unit, which is a research unit of the University of Witwatersrand, as well as the South African Medical Research Council. The Faculty of Health Sciences, where we're based, have been rearranged a bit, so we no longer fall into any specific department, but we have also recently been associated with the Infectious Diseases and Oncology Research Institute that is being established at the Faculty of Health Sciences. And it is here at the Faculty of Health Sciences that we are based and where we have been doing our research. We, as you can see from the name, the Antiviral Gene Therapy Research Unit, we have been doing a lot of work on developing gene therapy, and we have been working on gene therapy against viruses, specifically the hepatitis B virus. And the technologies that we have developed for our gene therapy, one of these technologies is in vitro transcribed RNA, which has become very popular in recent years due to the COVID-19 pandemic. And I'll explain a little bit about that in due course. To get into the talk, first of all, what exactly is in vitro transcribed RNA? What exactly is messenger RNA? Messenger RNA has, RNA of course stands for ribonucleic acids, and RNA has been discovered decades ago. And the existence of messenger RNA was actually first described in the 1960s, but its actual role in biology was not very well understood. Now, of course, after decades of research, we know exactly what mRNA does. We know, for example, that you start off with double-stranded DNA. All organisms and some viruses have double-stranded DNA or single-stranded DNA. And from that, they are able to make RNA through a process called transcription. So transcription, much like you would have transcribing of a meeting, you're going from one language to the same language. DNA and RNA are essentially the same language. They are both nucleic acids, so they are the same thing, and therefore you transcribe RNA from DNA. When you're going from RNA to protein, on the other hand, that's called translation, because you're essentially going from one type of language into a very different type of language. You're going from nucleic acids to amino acids, and that is what makes the proteins. So mRNA plays this very important role because it is the message between DNA and protein. Again, DNA is transcribed into RNA. One of those RNAs is the very important messenger RNA, which is translated then into a protein which will have a function. For example, we have hemoglobin in our blood. It's very important to carry oxygen from our lungs to the various parts of our bodies. And that is a protein. So we have the gene for hemoglobin, which is transcribed into the mRNA for hemoglobin, and that is eventually translated into the protein hemoglobin, which then has the function of transport of oxygen. So mRNA is very important. It's very important in producing these proteins. So once people understood what mRNA is, the application of mRNA, of in vitro transcribed mRNA, in the clinic was immediately obvious, because typically disease is caused by something going wrong. A virus is present, it is doing things that it shouldn't be doing, you get sick, or a protein is not present. People with genetic diseases, for example, that may not produce a protein. So if you can introduce an mRNA that has a therapeutic effect against the virus, or an mRNA that then produces the protein that is missing from that individual, you will then have clinical benefit. So using genetic sequences, using DNA and RNA, the application was immediately evident, and it has a long history of being used in research and development, as well as in medical application. So in vitro transcribed mRNA particularly really just refers to making mRNA in a test tube, in some sort of outside of a living system. And when you do that, you can make a sequence that looks like mRNA and behaves like mRNA, produces a protein that might have some sort of benefit. And this was, like I said, there's a long history. Since the early 1970s, actually, in vitro transcribed mRNA has been explored as for its clinical application. But it's actually really only taken off during the COVID pandemic, because making COVID, or rather making mRNA vaccines against COVID, against SARS-CoV-2, was one of the strategies that the people employed to produce an effective vaccine. And it just so happened that that actually the Moderna mRNA and the mRNA vaccines proved to be very efficacious. So I'm sure you've heard of the Moderna mRNA, as well as the Pfizer mRNA. And here in South Africa, actually most people probably have been inoculated with the Pfizer vaccine. and that's just the mRNAs, and they worked extremely well. And in fact, it was the Moderna mRNA of all the different mRNA, the Moderna vaccine of all the different vaccines that was the first to receive FDA approval, and that is really telling of how well this technology works. Further, the clinical application of in vitro transcribed mRNA also has made it very useful for the characteristics of nucleic acid-based therapeutics as well as prophylactics, that is vaccines, are very useful for clinical application because they can be scaled up very easily, or much more easily than traditional ways of doing things. For example, most vaccines in the market have typically been what are called subunit vaccines. So it is typically a protein or some small piece of a virus or a bacterium that is produced, but that is very difficult to manufacture. Whereas in vitro transcribed mRNA, you have fewer moving parts, and so you have a much easier to make this. It also has a good safety profile. You can regulate dose precisely, how much RNA you put in. You can get a very good indication of how much protein will be produced from that. There's no genotoxic recombination, and this is particularly problematic for, say, things that uses viruses. The other vaccine that also was used during COVID was the Oxford vaccine and the Johnson & Johnson vaccine. And you may know from media that is an adenovirus vaccine, which is a DNA virus. And so the virus gets into your cells, it produces the protein, and then you have a vaccine, an immune response, and so it works well as a vaccine. But you have the possibility that that DNA that is introduced can interact with your DNA, and that's what is known as genotoxic. If it recombines with the DNA in a particularly deleterious area, you can have very severe genotoxic effects. It's also very efficiently delivered. mRNA, for whatever reasons, for its chemical characteristics, it's actually very easy to get that into cells. So as I said, with RNA, it allows all of these are very easy to achieve these very important points that you need for a good therapeutic, as well as for a good prophylactic, for a good vaccine. So what exactly is in vitro transcription? Well, first it was developed early in molecular biology, as soon as, well, actually before people even knew what messenger RNA did, they were already working out how to make RNA. So making RNA has been known for a long time. And I'm going to be talking more about the in vitro transcription, which still use biological components. You can, of course, synthesize RNA in vitro, you can synthesize DNA in vitro, but the expense associated with it, certainly for making vaccines and making therapeutics, makes it very prohibitive. So in vitro transcription is certainly the more cost-effective way of doing things. So in vitro transcription, basically you set up a system that recapitulates the process of transcription outside of a living system. And so you have very few requirements. First of all, what we use is called a bacteriophage RNA polymerase. So bacteriophages are just viruses that infect bacteria, and an RNA polymerase is just an enzyme that makes RNA. So it's a polymerase, it makes polymers of RNA. You need template DNA because if you think back to the central dogma that I showed at the beginning, you need DNA to be transcribed into RNA. So you need to provide the DNA that is going to be transcribed into the RNA. And then you need the components that actually make up RNA, and that's the individual ribonucleotides. They are triphosphates. So in the case of RNA, you have guanine triphosphate, adenosine triphosphate, uridine triphosphate, and cytidine triphosphate. And these are put together into a polymer, and that polymer makes the RNA sequence. As I said, we use bacteriophage RNA polymerases. Bacteriophages, again, are viruses that infect bacteria. They're very simple. They have very simple components. And in this instance, the polymerases are actually very useful. In molecular biology, we use many other types of bacteriophages. But for in vitro transcription, there are three that are important, and that's the T7 bacteriophage, the T3, and the SP6. So their RNA polymerases are very convenient to use because you have a single enzyme, this protein, and it does basically everything, almost everything you need for transcription. And of the three, the T7 RNA polymerase is by far the one that is used the most often. So bacteria, we use the RNA polymerases from these bacteriophages. They are single DNA-dependent RNA polymerases, and that just means they use DNA to make the RNA. So again, DNA serves as a template for transcription of RNA, and they carry out much more complex transcription is very complex. We are eukaryotes, bacteria are prokaryotes, but in our systems, eukaryotic transcription is a very complicated process. So these bacteriophage polymerases, RNA polymerases, are able to recapitulate that very complex process in a test tube in a very simple system. It is a process of enzyme, meaning it will bind to the DNA and it will just churn out a lot of RNA. It will bind and it will will bind, add all the successive nucleotides that it needs to add. It does not dissociate it from the DNA. And so in a test tube, and in vitro, you can actually produce lots of RNA. And that's very important again, because if you want it to be clinically relevant, you need to be able to produce enough for everybody that needs it. And again, if you think of the COVID pandemic, the idea was to essentially vaccinate everybody, or as many people as possible across the world. And so you needed a lot of the active ingredient, the drug product. Another important thing about these polymerases is that they actually recognize a very short DNA sequence. This is what tells them start here and bind here and start transcribing. So it's very convenient that it has a short promoter sequence, because then you don't have to have a whole lot of extra DNA sequences that could make it problematic. The other thing is that it actually also transcribes very little of the promoter. In fact, it only transcribes the last nucleotide of the promoter. That is in contrast to eukaryotic transcription systems, which actually transcribes a very large part of the promoter. So these would then add unnecessary sequence to your mRNA, the mRNA that you want to produce. So for it to make very little of the promoter is very useful. Here, for example, is what the promoter sequences look like. So as I said, you start off with DNA, and that DNA is recognized by the RNA polymerase. So here you have the T7 promoter, the T3 promoter, and the SP6 promoter. So the T7 promoter will be recognized by T7 RNA polymerase. It will very specifically only recognize the sequence and will start transcribing. So that is the last nucleotide of the promoter, and it will be the first nucleotide of the RNA that is transcribed. So it reads this bottom sequence, and it basically makes the top sequence. So it reads it reads a C, it adds a G, it reads a C, it adds a G, it reads a C, it adds a G, it reads a T, it will add an A, and so on and so forth. So it reads the DNA, and it makes the complementary RNA sequence. So if you know what the RNA sequence you want to make, you simply design the DNA sequence that will make that RNA. You set up the system, and you have transcription happening. Excuse me. So as I said, you need a DNA template, but it actually needs very little. You just need the promoter sequence, depending on whether you're using the T7 promoter, T3 promoter, or the SP6 promoter, and that is then followed by your sequence of interest. And this is sufficient to initiate in vitro transcription. As I mentioned, it's a process of reaction. So what that means is when you have a promoter, the polymerase will bind, it will read the DNA, which is indicated in black here, and it will then produce the RNA. It will transcribe the RNA. And so it will just bind once and move along the sequence until it reaches its normal signal to stop. We don't use a specific sequence to stop, because that would require a whole lot of other things. So what we simply do is we use a linear sequence. So a linear sequence will have a defined end. So what happens then is when the excuse me, when the RNA polymerase binds to the promoter, it will transcribe the DNA until it gets to the end, and it will simply fall off. So you define the start by the promoter, and you define the end by using just a linear sequence. So these can PCR amplicons. PCR is a method to use to produce very large quantities of DNA, or a linearized plasmid. Plasmids are circular DNAs from bacteria and very useful to make DNA. So if you want to make a particular DNA sequence, you typically would use a plasmid to create that DNA sequence for you. But that means that the plasmid, since it is linear, since it is circular, it actually needs to be linearized, because you need to define the end where transcription will stop. So in vitro transcription of messenger RNA, at least what we want to do is we want to make a eukaryotic mRNA. We want to make an RNA sequence that when it gets into a eukaryotic cell, so that is into our cells, it will actually then be recognized as an mRNA and be translated by our cellular machinery into a protein. So there are a few things that you need. The eukaryotic mRNA typically has what's called a 5' cap, which is just a methyl guanosine that is added to the RNA in an inverted orientation. You have an untranslated region, so there's part of the RNA that is not converted to protein. You have the open reading frame, and that is the region that is converted to protein. And you have the 3' untranslated region, which also is not read for making it into protein. And the 3' UTR also contains a poly-A tail, which serves a very important regulatory role in these mRNAs. So basically what you would do is you would produce a DNA template, again, for example, a linearized template DNA that will have all the components that allows that when you do transcription, it will create a sequence that looks like a eukaryotic mRNA. So you will have the promoter that will drive transcription. You have the 5' untranslated and the 3' untranslated regions, as well as the open reading frame. So that produces the RNA, and when this RNA gets into the cells, translation will basically start at this point. It will read the RNA sequence, it will stop at that point, and then produce the protein of interest. So for example, in a vaccine, like the COVID vaccines, we have mRNA, the Moderna vaccine and the Pfizer vaccine it contains the RNA for the coronavirus spike protein. So when the RNA is introduced into the cell, it is essentially translated to produce the spike protein. And when you produce a spike protein inside cells, eventually the body will mount an immune response to it, and that is essentially how you develop protection against the full virus. 5' capping, however, cannot be encoded by the DNA, so it has to be done separately. In eukaryotes, this is basically how it would work. So you have a triphosphate, which is usually the first one that is added, and during transcription you have a second nucleotide coming along. I'm of course not showing the DNA, but a second nucleotide would come along. You have a phosphodiester bond created between excuse me, the 3' hydroxyl group over here and this 5' carbon over here, or the 5' phosphate to be more specific, to create a phosphodiester bond. A pyrophosphate is released as well as a proton, and you now have this phosphodiester bond to first start the chain. This is repeated over and over. Another nucleotide comes along, a triphosphate, have a phosphodiester bond being created, and this is repeated to make the entire chain. And for example, the chain for the spike protein of coronavirus is about 3,000 nucleotides long. So you have 3,000 of these bases in a particular order. Now, capping actually first entails removal of one of the phosphates by a dephosphorylation step. So you have an enzyme called an mRNA triphosphatase that will remove that phosphate group. And then you have guanosine being added. So guanosine is a nucleotide, and it is added pretty much the same way as a phosphodiester bond is being made, but it is added to create a 5' to 5' triphosphate bridge in this instance. So again, you have a pyrophosphate removed and a proton removed, and so now what you have is a cap, and this is called cap naught. There's a modification that happens to it as well. What you can see is at this N7, you have a methyl group being added to form the full cap naught structure. So this is very important. Without this, the mRNA will not be translated. The machinery will not recognize it and translate it. You could also further have methylation of the first base to produce cap1, and methylation of the second base to produce cap2. And this basically produces the RNA sequence that you can now use and administer, produce for vaccine or for a therapeutic. So the 5' cap is very important. Normally, RNA is produced, at least in our systems, in eukaryotes, RNA is produced in the nucleus, and it needs to get to the cytoplasm where it is translated. So it's very important in nuclear export. It's very important in preventing degradation from the 5' end, because the RNA needs to be stable for as long as it needs to be translated. But very importantly, it is very important in translation. That 5' cap is actually recognized by the cellular machinery as a message, and that needs to be translated. So the machinery will bind to this 5' cap and then start the translation process. It's also actually very important in innate immune responses, but I will not go too much into that. And it's but just to say that it's very important for that to be there, for it to be functional to work very well as an mRNA. Alternatively, instead of so that is how it works naturally. But what you could also do is a in vitro capping by using a cap analog. So these are dinucleotides. Typically, there are two nucleotides bound together to basically produce the two guanosines that are joined by a triphosphate bridge. And I'll mention some of the trinucleotide analogues that are used as well. So usually there's a requirement for the first nucleotide to be a G, and that basically comes down to the different promoters. So if you look again here at the T7 promoter, the last nucleotide in the T7 promoter is a G. Therefore, the first nucleotide of the RNA will be a G. And so that is basically what is used. The cap is also a G, and therefore the dinucleotides, the analogues, are basically two guanosine nucleotides next to each other. So basically, it looks something like that. As I mentioned, the cap is inverted relative to the rest of the RNA, and so analog is in the first instance. Then you will have this essentially what looks like a capped RNA, and that then of course is the transcription is continued as normal to produce the full RNA. But now, since the first nucleotide that was added was the analog, you basically have your RNA having a cap. As an alternative to analogs, there's also enzymatic capping, which I will get to just now. So this is basically what the cap analogs look like. As you can see, there's a guanosine over here, and there's a methyl guanosine over there. But the problem is that normally this is where the next nucleotide will be added, and this is where you want it to be added. But this hydroxyl group basically is very similar to that. It's also at the 3' end. And so you can also actually have extension from this end, but that creates a non-functional mRNA. So furthermore, people have created what are called anti-reverse cap analogs by basically modifying this hydroxyl group, adding a methyl group to it, so that now you can only have, you cannot have the transcription extending from this end, rather you can only have transcription extending from this end. And so the mRNA produced from that is a proper mRNA that has a proper N7 methyl guanosine cap. I mentioned there's also trinucleotides now. A company called TriLink produces what they call clean caps. And as you can see here, you have a trinucleotide. So there's a G and a G, and over here we have the a guanosine cap. Also, you can have an A and a G with a guanosine cap, or an A and a U with an A and a U with a guanosine cap. And so these work very similarly, and that they will be extended from this point onwards during in vitro transcription. And these work exceptionally well for making functional mRNA. So that is what's called co-transcription capping. You add the analog during the reaction, and it will be added as a cap. Alternatively, what you can do is use enzymatic capping, where we use from a virus called the vaccinia virus. It again has a single enzyme that carries out all the different activities of capping. It has a triphosphatase, which is the removal of the first phosphate, has a guanylyl transferase activity, which transfers the guanosine onto the first nucleotide through that and creating that 5' triphosphate bridge. And then there's a guanine methyl transferase activity, where it adds a methyl group to the N7 of the guanosine, thereby creating a cap. And so that cap that is produced, that RNA that is produced, will now have a 5' cap. It's cap naught. You can add a second enzyme, a methyl transferase, to add a methyl group onto the first nucleotide, thereby making cap 1. And these can then be used for in vitro transcription. To make the in vitro transcribed RNA, which can then be translated. Just to briefly mention, there are 5' and 3' UTRs on the DNA and on the RNA. And these are usually very important for regulatory roles. They're important for initiating transcription, for initiating translation, as well as producing good amounts of protein. In the 5' UTR, you also have the Kozak sequence, which is essentially very important for where translation starts. It's important to ensuring that you don't have premature decay of the mRNA, and allows long-term expression of the mRNA. And often the UTRs from the globin genes. So as I mentioned, for example, hemoglobin is made up of a number of globins, and hemoglobin you need in a large quantity. So the UTRs from globin mRNAs typically are very good at producing large amounts of proteins. The poly-A tail is also part of the 3' UTR, and it's very important in translation. It's usually either added enzymatically or it is encoded within in the template. But it's very important to have the stretch of A's, at least 15 nucleotides to allow efficient translation. And what it also does is it prevents degradation from the 3' end. So the longer the tail, the less likely there is going to be degradation of the RNA from the 3' end. Just to mention that mRNA is inherently very unstable, and so also what we do is we incorporate nucleotides that have modifications, and so these modifications are very important in ensuring that the RNA is not degraded prematurely. So here, for example, you see all the different types of modifications that are available. So there you have adenosine, and then you have N6 methyl adenosine. So it's just an adenosine with a methyl group over there. You have N1 methyl adenosine with a methyl group over there. Similarly, you have guanosine modifications, cytidine modifications, uridine, as well as uridine modifications, and pseudouridine, methyluridine, methoxyuridine are all modifications of uridine. And the important one I want to point out is N1 methyl pseudouridine. So this has been chemically synthesized to have this modification. So it is basically you may notice that uridine and pseudouridine are just mirror images of each other, and pseudouridine and methyl pseudouridine are different by this one methyl group. And the Moderna vaccine, as well as the Pfizer vaccine, actually contain N1-methylpseudouridines, and that is what helps them be very stable, improves the translation. You get lots of protein from it, and so you actually get a very good immune stimulation, which is of course important for vaccine development. Finally, just to mention, the mRNA vaccines have, or in vitro transcribed mRNA have numerous advantages over more traditional vaccines that we have used, and you can see that clearly from the pandemic. The Pfizer vaccine and the Pfizer vaccine, the Moderna vaccine have been the vaccines that have been used most frequently. Again, it's much more simpler to produce and purify these than subunit vaccines or virus-based vaccines. So that of course means that it is much easier to adopt this technology and use it for vaccines and therapeutics. An important point is that the antigen is expressed. I'm not going to go into too much detail, but except to say that we have two arms of the immune system: the cell-mediated immunity and humoral immunity. And usually vaccines produce a very strong humoral response, so we get very good antibodies against the vaccine. But with mRNA vaccines, you also have a T-cell response, and that may actually be why they work so much better. There's ease of delivery, there's rapid response. I've mentioned that they deliver it very easily. Rapid response because we only need to deliver them to the cytoplasm, so they get into the cytoplasm, they encounter the cellular machinery, and they translate it. And they actually have a short half-life, which can be a disadvantage, but I've mentioned how it's possible to using modified nucleotides to actually stabilize the RNA and have it expressed, have a much longer half-life, and so work much better. Finally, I just want to give a few acknowledgements. This is Professor Patrick Arbuthnot. He's the director of our unit. That's also my colleague Christy Bloom, and we have been working a lot on what you may have read about the mRNA vaccine transfer hub. It's a global initiative by the WHO. They've recognized South Africa as the site where this hub is being developed. So in Cape Town, there is a company called Afrigen, and working with Afrigen to get mRNA technology up and running. So we are working with them, and we've had some quite good successes in making an mRNA vaccine, a COVID vaccine, and we've gotten some very nice results in mice to show that these vaccines work really well. That brings me to the end of my presentation. Thank you, everyone.
Thank you so much, Professor Abdullah. We are now going to move to the Q&A session of this presentation. If you don't mind also turning on the camera, so that way we can have a conversation. Thank you so much for your presentation. I have quite a few questions. Okay, so the first one I want to ask, you mentioned the short half-life of the vaccine. Does this mean mRNA vaccines need to be taken multiple times in order to provide continuous protection?
It's an interesting question. So it depends. A vaccine, of course, it depends on the. It doesn't work like a therapeutic. It's not like you need to have it constantly there. You just need it to have that initial response. You know, so it's much like any other vaccine that you've taken. If you take flu is not a good example, but if you take the flu vaccine, that basically protects you for that entire year until a new strain comes around. So you just need that initial response. For a therapy, if the virus is along for a long time, you need that mRNA to be there constantly producing the protein to have a therapeutic effect. But for a vaccine, you don't need it really you don't really need it for a very long time. You just need to get it. You just need the RNA to get in, the RNA to be translated into a protein, and then that protein will elicit an immune response. So once you have that immune response, you basically have that immune response essentially for life. You may need boosters, and so again with COVID, there's often how you need to go for a booster shot, and so you need that booster, and it's usually it will be irrespective of whether it's an RNA vaccine, a DNA vaccine, a virus-based vaccine, a protein subunit vaccine. You will need that booster shot after a while. The good thing about the mRNA is that you can actually administer it multiple times. If you think about the Johnson vaccine, so that was the first one that came to South Africa, for example, and that was usually the one given to medical professionals. They got a virus vaccine. So when you administer the virus, while the virus does produce the protein that we want the immune response to against, your immune response, your immune system actually also generates a response against the virus itself. So you cannot re-administer that virus, and you need to actually administer a very different virus. So that's a limitation of using, say, a virus. But with the mRNA vaccine and with the vector that it uses, you can actually do it multiple times.
Okay, that's very interesting. Thank you. Talking about mRNA vaccines, why is mRNA the RNA that is used for these vaccines and not tRNA or rRNA?
Okay, so that comes again down to that central dogma that I spoke about at the beginning. So DNA is transcribed into RNA. It is the message that is read on the DNA, and that you make the RNA. So DNA you can have many different types of RNAs. We have different types of genes. We have genes for mRNA, we have genes for transfer RNA, we have genes for ribosomal RNA. There are more RNAs being recognized almost on a daily basis. Small interfering RNA, microRNA. But of these RNAs, only messenger RNA are called coding. All the other RNAs are non-coding. So they do not actually produce a protein. And for a vaccine, you need to have a protein made, because you need to have the immune system recognizing a protein and mounting an immune response against that protein, or a very complex molecule, but usually it is a protein. So ribosomal RNA are important in translation. They are important in they have function in translating RNA, but the ribosomal RNA themselves are not translated. Transfer RNA as well, they transfer the amino acids to the site of translation to make the protein, but they are not translated themselves. So it has to be an mRNA, because the mRNA is actually what gives the message to make the protein, and it is that protein that will have the function. And that is, although that is of course then only important for vaccines, because again, for the vaccine you need to have some sort of protein. The introduction that Walid gave mentioned some of the work that I did in my PhD, which was using the RNA interference pathway. In that instance, the RNA interference pathway just uses RNA. It does not. It is the RNA itself that has a function. In this case, which has a therapeutic function. So if you're making an RNA from the RNA interference pathway, then it will be a different class. It would be a class called short interfering RNA or microRNA, because there you do not need translation of the protein. You just need to make the RNA, and it's that RNA that has the function.
Ah, okay, that's interesting. Another point that you brought up, so the mRNA gets transcribed into the genetics. Is there a possibility that natural mutations in a cell could mutate that mRNA sequence, rendering the vaccine maybe null and void?
So usually our RNA is normally modified in a way, those modifications I spoke about. So when it makes the RNA, when it transcribes the RNA, you can have it uses just the normal nucleotides. Is used after it has been transcribed. Then we have again numerous systems to modify the RNA to make, for example, the N1 pseudouridine. Those happen within the cell. mRNA vaccine is introduced, it is introduced in such a way that it does not encounter. So you would not expect that type of modification to become inactive. There are other immune regulators that actually are present in cells that do exactly that, and they might be they could potentially, though I don't know of much research being done with vaccines, but they potentially could do that. And in fact, for example, the reason why HIV is thought to be much like COVID, is thought to be a zoonotic disease. It jumped from primates to humans, but in primates, their immune virus is actually not as deadly as HIV, because their immune system actually modifies the viral RNA so extensively that it is not functional anymore. In our case, HIV gets into our systems. There are many other ways of limiting it, but this particular way, this RNA modification pathway, does not modify it sufficiently to make it inactive. But whether that pathway could potentially function on vaccines has not been, to my knowledge at least, has not been studied.
Okay, thank you so much. Another question I have, you mentioned a collaboration with AfricaGen. When do you think Africa will have the capacity to have its own center for manufacturing mRNA vaccines?
So that's exactly what AfriGen is trying to do, and that's what the whole initiative by the WHO is. And the initiative is that the AfriGen will be established as this hub. So it's called a hub because they employ what they call the hub and spoke model. So AfriGen will be the hub, and a number of spokes have been identified. There's a spoke in Egypt, India, Indonesia, Brazil, Argentina, Bangladesh. So all of these low to middle income countries have been identified. AfriGen is going to be able to, AfriGen is establishing themselves as the hub to be able to set up manufacturing, and they will then transfer the knowledge to these other hubs. Sorry, spokes. The first spoke that they will transfer the technology to is called BioAC, and BioAC is a vaccine manufacturing company also in Cape Town in South Africa. So the idea is that AfriGen will establish large-scale manufacturing capabilities and then teach all the spokes. And the plan at the moment is for this to be mid next year that they will have this set up.
Okay, that's lovely. So very soon then. That's excellent.
Yeah. So I mean, it's a lot of work, of course, and it's we are helping them in setting up all of this. But yeah, like I said, so far we've actually had some very good results showing that we can actually make a vaccine very much like the Moderna and the Pfizer vaccines that actually produces an immune response from our mRNA.
That would be excellent, especially for countries that can't afford to pay for the fees from Moderna and Pfizer and whatnot.
Exactly, and it's very sad that Africa, for example, imports like 99% of its vaccines. Africa only manufactures 1% of its vaccines. So the idea is that we establish this technology and transfer the technology then to other low and middle income countries and establish the technology there as well, so that we can make these vaccines ourselves.
Yeah, I agree. Pivoting towards your work, I see you have focused a lot on hepatitis B research. So would it be possible to use mRNA vaccines to cure hepatitis B?
So everything is very different. You know, a cure is, you know, cure can be very difficult. Very much like how you have a difficulty finding a cure for HIV. HPV is hepatitis B is similar in that once the virus gets in, it's very difficult to get rid of. So people tend to talk about a sterilizing cure and a functional cure. So a sterilizing cure will actually be something that eliminates the virus completely. A functional cure is basically eliminating the disease, but not necessarily eliminating the virus from your system. So for hepatitis B, the more likely goal or more achievable goal is in the field is considered to be a functional cure, not a sterilizing cure. There's work towards therapeutics, and using an mRNA, we can potentially get a therapeutic. But the vaccine obviously tends to not be therapeutic. Vaccines tend to be prophylactic, and that they prevent disease, not really cure disease. So you are given it the vaccine before you contract the disease in the hopes that you do not contract the disease or contract a milder form of the disease. Using mRNA, using DNA is extensively being researched as a vaccine. But interestingly, the data actually suggests that these could potentially have a therapeutic effect, even though you design them as a vaccine, which tends to be prophylactic. There is the chance that they will actually be therapeutic, and that is the one point I mentioned to the end of my talk about there being two major arms of the immune system. You have the humoral response, which is what produces the antibodies, and you have the cell-mediated response. Typically, vaccines only induce the humoral response. You may have heard also in the media about how the COVID vaccine produces antibodies because you need those antibodies to protect you. But T cells also protect you. And if you can get a T cell response, you could potentially get a therapeutic response as well. So yeah, you could potentially get a cure, but that's all still extensive, still much being researched.
Hopefully you'll find something as well soon.
Yeah, so our research is into other areas as well. Like I've kind of limited my talk to mRNA and for prophylactics, but there are many different types of potential therapeutics that you can also use. So if you produce an mRNA if you make an mRNA that produces a protein, and that protein is designed to have a therapeutic effect, it could be used to cure many different diseases.
Interesting. I just have two more questions left due to time constraints, but I am intrigued. Is it possible to solve other diseases with mRNA vaccines besides viral diseases? So things like bacterial or parasitic infections, for example.
At least theoretically, and theoretically, you could have vaccines against many different diseases, against other parasites and bacteria as well. And again, with this whole mRNA hub that the WHO has is initiating, there's a very important component of it where they want active research, because what we are doing currently is we are making a vaccine against COVID, and COVID hopefully is not going to be around forever. So we can't just set up all this capacity for making COVID vaccines. We need to be able to make other vaccines as well. And so there is a consortium that has been set up to look into other targets. And one of those targets is actually the TB, the pathogen that causes TB, which is a bacterium called Mycobacterium tuberculosis. So in that instance, we want to see, can we actually make a vaccine that protects you against TB? So the short answer, theoretically it is possible. We will have to do the work to see if it actually works.
Fingers crossed it comes soon, because it definitely needs it.
Fingers crossed.
One last question. Do you know if there's any other uses for mRNA technology besides vaccines?
Yeah, so that's the therapeutics that I've been talking about. So again, vaccines technically just means that it has a prophylactic effect. You administer it to prevent disease, but mRNA technology, of course, can also be used for for a therapeutic effect. So as long as the protein you produce, as long as the protein you produce has some sort of therapeutic effect, it has potential for therapy. So take, for example, in people with diabetes. Often people with diabetes require insulin. Insulin is a protein that is produced from an mRNA. Now, what normally happens is we actually produce insulin, and people take insulin injections. People with diabetes take insulin injections. So you could theoretically, instead of giving them the protein, you could give them the RNA that is translated into the protein. And similarly, you could take it a step back. You could give them the DNA from which the RNA is made, from which the protein is made. So that is an example of how you could use mRNA technology or even DNA technology for therapeutic effect. As long as it produces some protein. One of the things that we are working with in our unit is you may have heard of genome editing technology. Genome editing technologies are proteins that will bind to DNA and actually cleave it. When they bind the DNA, they cut it and thereby inactivate the DNA. So take, for example, HIV. Once it inserts itself into your DNA, it is DNA. It inserts itself into your genome. It is DNA. Similarly, with hepatitis B virus, it's a DNA virus. So we have actually made these proteins that will target the virus. And what we do then is we can either make the DNA that will make the RNA that then makes the protein that targets the virus, or we just make the RNA that's translated into the protein that targets the virus. So as long as it produces some sort of protein, you can use the mRNA for that.
That's excellent. Well, thank you so much for joining us as our speaker, Professor Abdullah. Thank you for answering all these questions. I wish you luck with your hepatitis B research. I'm now going to pass it on to my colleague, Walid, to do the closing. Thank you very much.
Thank you. Thank you. Sorry, Walid, I can't hear you. I think you're on mute.
Yeah, sorry, technical problem. I would like to thank Professor Abdullah for accepting our invitation and for giving this amazing presentation and answering all these questions. Also, we would like to tell our audience that we have an IYCN workshop series on professional development. You can find it on the IUPAC YouTube channel. We also made our first IYCN Science Café in July. You can also find it on our IYCN YouTube channel. We would also like to announce that we are going to have another, our third IYCN Science Café on November, so stay tuned. We would like also to tell that we are committed to equity, diversity, and inclusion, and won't tolerate any discrimination towards our members or toward anyone. Lastly, I would like to thank again Professor Abdullah for his talk and all the attendees for attending this Science Cafe, and thank you all. You can all you can follow us on all social media or visit our website iYCNglobal.com for upcoming events. Thank you.